The next panel I'm deeply excited about. It is the science panel, and it highlights scientific research on mini-forests, covering topics such as their cooling benefits, insect-host plant relationships, microbial and fungal communities, and data collection methods in the Northeast US and Belgium. We'll hear from Dr. Nicolas Geron, Nicolas de Brabandère, Prassede, Calabi, and Melory, Brandao. Their presentation will be followed by a Q&A, so please leave your questions in the chat. And now I'm going to read their bios really quick. And I apologize for my Romanian accent in advance. Dr. Nicolas Geron is an assistant professor of geography and sustainability at Salem State University. His research focuses on remote sensing, drone applications, urban forestry, and climate resilience. He is especially interested in how Miyawaki forests can help mitigate extreme heat and uses remote sensing technologies to study the climate impacts of urban forestry projects in Massachusetts and Pennsylvania. Nicolas de Brabandère is a biologist, nature guide, and the founder of Urban Forest, a Belgium-based company that specializes in the creation of Miyawaki forests and biodiversity gardens. Since 2016, Nicolas has contributed to the creation of over 130 Miyawaki forests in Belgium, France, and Germany. Nicolas has also published several papers on the Miyawaki Method and data collection in these forests, contributing to the field's growing body of knowledge. Prassede Calabi is the founder and project director of Windfast Forest. With over 30 years of experience, she has taught academic and professional workshops, served on grant committees and advisory boards, and contributed to numerous education grants. Her peer-reviewed publications span animal behavior, ecology, and pedagogy, reflecting her deep commitment to both science and education. And lastly, Melory Brandao is a graduate student in wildlife ecology in Dr. Doc Talamy's lab at University of Delaware. With a background in ecological restoration, her current research focuses on insect communities in Miyawaki Forest in the Boston area. She's studying how different planting methods influence insect biodiversity, herbivory, and overall urban ecosystem function. And the order in which we are going to go today is Nicolas Geron, Nicolas de Brabandère, Dr. Prassede Calabi, and Melory Brandao. And if you have any questions, please put them in the chat. And I think we're ready to start. There's other scientists that couldn't be with us today, so I'm hoping at some point we can do a larger gathering to exchange knowledge and activate new knowledge. So I'm excited about that.
So yeah, thank you for the introduction. My name is Nick Akira Geron. My middle name is actually Akira, and when I saw that Dr. Akira Miyawaki had a similar namesake or the same namesake, immediately jumped out. But not only that, I was excited to look and learn about Miyawaki forests after spending my PhD studying tree planting. So I'm going to be talking about using some initial results on the Danny Park Forest. You can see on the image on the right, the Danny Forest, the Miyawaki Forest is right in the middle. It's a circle. And I'll be talking about the forest as well as the surrounding grasses, larger plants, mature trees in the park, and trees that are along the pathway as well. My background, I just want to be clear, as a geography professor, so ecology is not. I've learned a lot in the yesterday and looking forward to learning more today about the ecological systems. But I really study the human and environment interactions. So how people interact with trees and how trees are faring on public streets as well as in private backyards. My dissertation was around the Greening the Gateway Cities tree planting program. I've looked at the impact and success of trees, like this cherry tree in Worcester or this river birch in Fall River. And that's how we measured the height of trees when they got really large, is to stick a 10-foot pole on top of our heads if the tree was larger than 10 feet. And since these trees, we've actually just been back to a few of these cities this summer, and some of them have grown over 20 feet. So we now have to use a hypsometer to measure them. They're too tall. But that's my background. And all the time that I've spent in cities, especially in environmental justice cities or post-industrialized cities that are very dense and have very little space for planting, as Doug is saying, an individual tree just doesn't make that much of a difference, especially on a street. And so I was really excited to hear about the Miyawaki forest and this idea of having patches of trees of very dense, also native vegetation. So this is the forest that I'm going to be talking about. It's the first forest that was planted in the Northeast. And when we flew the drone in 2024, we measured trees that were 21 feet tall, and they were only 3 years old. So quite impressive. And this is going to be looking at thermal imagery over the course of a single day. So the questions guiding this initial research are, what is the land surface temperature of these dense patches of vegetation, and how do they compare to the single trees that are along in the park, along the sidewalk, as well as larger mature trees that were planted before, and then trying to understand that both through the raw temperature, but also through standardized z-scores or relative temperature. So relative temperature allows us to compare temperature throughout the day. So we can compare 11 a.m. To 7 p.m. And I'm going to be presenting the results of the 3 flights. This was on Labor Day in September last year. We flew at 11 a.m., 3 p.m., and 7 p.m. You'll notice that the flights were all conducted at 100 feet elevation or altitude, and that this was a pretty typical summer day, 78 degrees Fahrenheit, but not an extreme heat day in any sense. And on the right, you can see my daughter in the double stroller and the drone. I have some very helpful field assistants. I'm not going to get too much into the methods, but this could be useful for anyone looking to replicate this or do similar studies. So I'll just say that the images had to be georeferenced, and then we manually drew out the different land cover classes in the area, and then did 100 randomized points within each land cover class. And then once we had the land surface temperature, we were able to calculate the relative temperatures. And so this is some of the initial results. These are the raw temperatures in Denny Park. You can see in the 11 a.m. image on the left that the darker areas are the coldest. Those are probably also in shade. At 3 p.m., everything is heating up. You notice that the pavement in particular is extremely hot. The maximum on the legend here is 40 degrees centigrade, but this was often, in the 50s or 60s. And I do want to note that remind you that this is land surface temperature, so it's not air temperature. And then you can see in 7 p.m., everything is starting to cool down.
To do that container before the
in particular, the grass. So we found again and again that anything that was in shade in particular, especially if it was vegetated, was often the coldest in the scene. So what we're able to do with the randomized points is basically understand temperature change for these different land covers throughout a day. And you can see here on these graphs, different colors represent different land covers. So bare soil is that light purple. The Miyawaki forest is the red. And then temperature is on the y-axis. And the x-axis are the 3 different flights. So we have the 11 a.m. flight, the 3 p.m. flight, and the 7 p.m. flight. I'll talk about the mean land surface temperature graph first. There you can see the Miyawaki forest had the lowest average temperature. in the morning and at 3 p.m.
Anything?
when the day is hottest.
Yeah.
We also found interesting that the grass actually dropped to the lowest at 7 p.m. When we looked at maximum land surface temperature, this is where we saw the largest amount of variability between all these different land covers. And it was really exciting to see that the Miyawaki forest was the lowest temperature at 3 p.m. Or pretty consistently throughout the day. And you can see that, it's almost, it's 2 to 3 degrees lower at the maximum temperature. So wherever it's hottest, and you can tell that the bare soil and the grass and the pavement were about 20 degrees hotter maximum. And then also interesting was the standard deviation. Here you want to mostly pay attention to the amount of variance is what the standard deviation is measuring. And so we can see that this Miyawaki forest has the least amount of variance at each time of the day. So at 11 a.m., it's a very consistent temperature. At 3 p.m., it's a very consistent temperature. And at 7 p.m., it's a very consistent temperature. And that's great to see as well, just because it was often the colder temperatures on average and in the maximum. So it's consistently pretty cool. It was fascinating. So I think the high variation in the other land covers has to do with shade, as well as potentially like dead branches on larger mature trees and things like that. When we got to the relative temperature, this is where it got very interesting, and I'm still trying to understand or wrap my head around this. But essentially, this is a difference image. So this is from 7 p.m. subtracting 11 a.m. What we see is that relatively the forest is actually hotter than the surrounding area. And there could be a few different reasons for this, but my biggest suspicion is that if you look at the image on the bottom right, you can see that at 11 a.m., the grass is actually extremely hot. And then at 7 p.m., the grass is much cooler than typical. And so I think the Miyawaki forest, by not changing as much throughout the day, by being relatively consistent, it is cooler in the morning, but than the surrounding area, but it's less so. And so by the end of the day, it's changing, but it's changing slower than the surrounding grass, and is not as shaded perhaps at 7 p.m. As Alex said in the very beginning, these plants are part of the water cycle. And so there's a lot more water in the Miyawaki forest as well, and in these larger shade trees. And that could potentially be playing a factor, as water has a high specific heat. So it cools and heats up faster or slower than surrounding. So in conclusion, we found that the Miyawaki forest that's 3 years old was about 20 degrees cooler than the surrounding grass at the hottest time period of the day at 3 p.m. But more encouragingly is that this Miyawaki forest had a similar temperature profile to the mature trees and the street trees that were planted over 20 years ago. So in a very short amount of time, it has a very similar temperature profile to those much larger vegetation. We did find that the largest amount of cooling comes from shade. And so we do need to understand transpiration, and we're beginning to measure air temperature this summer. And then we are interested in the relative cooling and warming to the Miyawaki forest compared to the landscape surrounding it. And so the Danahy Park microforest was in a park surrounded by grass, surrounded by other vegetation. So we're also performing similar experiments with the Worcester Miyawaki forest, which is pictured here on the right, which is not surrounded by anything other than the parking lot, the road, the sidewalks. There are a few trees, but they're all very young. And so I'm working with Clark University as well this summer to measure the thermal or the land surface temperature during one of the more recent heat waves. Although today is another day that would have been excellent. But you can see here that these are, again, 1-year-old Miyawaki forest, but already there's about a 14-degree difference between some of the more denser patches and the pavement and the road. So I want to say thank you. Thanks. Really appreciate the opportunity to talk, and really excited about this conference. Thanks to my drone pilots, my two daughters who have been very, very, very understanding and have visited multiple Miyawaki forests. And big thanks to Biodiversity for a Livable Climate for hosting this and as a partner.
So I'm Nicolas. I have a training in biology. I got to know the Miyawaki method in 2015, like many people, through a TED talk by Shubhanda Sharma in India. And I started the first one in 2016. So that was the first one, the first experiment. And the results are quite stunning. Actually, we've never seen trees grow as fast as this in Belgium or even in Europe before. That's quite something. Let's skip through some examples. That's a site in the north of Paris, a very polluted site, and that's a result in 60 years. That's next to an industrial facility in 2 years. I also work for properties. Yeah, this one. I also work for public services. That's a park. We created this. This forest is along a road. I also work in schools. I work with a collective of citizens also. So far, I've done over 130 Miyawaki forests. I also work in a Mediterranean climate. That's quite interesting because all the species are different. It goes slower. It's very dry. And soon I'll be doing one in Greece on one of the islands. So that's quite exciting. So the context in which I operate, most of the time it's very degraded soil, very compacted, very little biology or oxygen in the soil. It's always on small plots of land that require very intensive regeneration with a lot of expectation from people, and it's mostly in urban areas. From my experience, there are really three main elements to remember. Soil preparation is key. You have to know what you're doing with the soil. Species selection is tricky. This concept of potential natural vegetation, you really have to dig into that and understand what it's about and not plant randomly. And maintenance is crucial for the first 2-3 years. And you have to find the right balance between, looking after the trees you've planted, but also, maintaining biodiversity on your patch. Potential natural vegetation is a scientific concept that started in Germany. It represents the dominant vegetation of a particular site, as if no human influence. It's constructed on remnants of past vegetation still present today. So you have to look for native forests in the same conditions, and that's how you can find, what potential natural vegetation is. It's different from actual vegetation. You have to explain that all the time. And it's interesting because that's the whole idea of Professor Miyawaki. It's really to compare the degree of deviation that we're witnessing because of progress and human impacts. He does take into account changes that have resulted from human impacts. And it's now used in order to define objective and restoration program. So this concept is really growing and gaining momentum. I study in depth 6 Miyawaki forests in Belgium in different locations. I look at the 50 square meter patch and actually map every single tree species. I took measurements, etc. I also present a report with photograph and simple indicators. So it's not research, that is published in a journal. It's done seriously for its own sake. So you can see the evolution of the forests. I show photographs because sometimes people have very strong opinion about Miyawaki forest, but they haven't even visited one. So it's a bit a way to force them to actually feel what it's like in there. So you see the whole forest, and you can really picture every single tree, how wide they are, what the ground cover is like. And you also have this profile with the different layers in the forest. And then I made lots of data. Most importantly, it's really survival rates. Perhaps you can compare proportion of trees and shrubs. I started planting many shrubs at the beginning, and by talking with Professor Fujiwara in Japan and Elgin Box in America, I realized that you really have to plant more trees and less shrubs. So all this data is available. An interesting study I did also is looking at the soil food web. So very simply, I took sample inside and outside the forest and look at it through the microscope. And I only noted, presence of absence of different. Oops, sorry about this. I have to switch this off. So what you can see is that the soil food web is way more developed inside the forest than outside. And when you look at it through the microscope, it's very obvious. You get bored. When you look at the soil outside the forest, you realize how much degraded soils are in urban areas. We also look at temperature, and like Nicolas said before, the temperature difference is quite dramatic, actually. And even in the photo below, you can see that the children are naturally driven to cooler areas. You can also compare forests, and you can see that they look very different. The ground cover by the colors, but also the density of the trees and all of that. It's quite interesting. And in profile, also you see massive differences in growth from side to side. Some are really miraculous, they go crazy, and some are very slow. It's difficult to understand why that is. It's not that simple to really get a good understanding of that difference. So the main finding, in Belgium, in temperate climate, the average growth is about 75 centimeters per year. That's about 3.5 feet every year. It's very quick. Mortality rate varies from 10% to 30% within the 6 years. And usually when the mortality rate is higher, it's because we're in schools and places like that where the children, play inside the forest, break branches and all of that. The Miyawaki method works extremely well, even on very degraded soil. It's extremely resilient to drought. In 2022 in Belgium, we had the worst drought in 50 years, and most of the sites were not, we didn't water, and it went fine. Of course, drought has slowed down growth. Trees become dormant after a while. Definitely maintenance is not required after 3 years. Despite very quick growth, we don't see stem collapse. The soil biology is significantly better inside the forest compared to other side. To me, this is the best indicator. It's the the water infiltration time, and we noticed that it's 6 times quicker in the forest compared to outside. And to me, this is a very good indicator because the faster water infiltrating the soil, the better your soil is. Temperature is much lower in forests. Sometimes proving this is a bit like, proving that, talking is good for communication. But anyway, and the concept of potential natural vegetation require in-depth appreciation and understanding. It's a tricky concept. It's difficult to grasp and understand truly. That's an example of a temperature map, just like Nicolas. So the place in the circle is where the Miyawaki forest is, and you see that it's cooler. Of course, water body at the center are cooler, and big trees are cool just as well as Miyawaki forests. Miyawaki forests don't do better. They just do the same service. That's biodiversity is quite challenging to monitor in Miyawaki forests, and that's a great study we're doing in France. It's a 10 acres land park with 12 habitat types. The Miyawaki forest is the brown color, and the idea is to study, does the Miyawaki forest have a positive impact on biodiversity, or does it remain the same? And we will look at it over 5 years. That's another study that they did in the engineering school that was quite interesting to do a Miyawaki forest, with engineers. And they actually look at noise level. So you see the road on the left. It's there's a lot of traffic. So they look at noise level. You can see the graph on the left is a noise level near the road, and the graph on the right is behind the forest. So you can really see that you don't get all the peaks, and it's definitely lower noise. So that's interesting, in places with properties, and schools and places like that. That's another, for a client is working on 4 sites, and they want to look at temperature, humidity, CO2, and different. So they have these monitoring devices inside and outside the forest, and they compare. So that's an example with temperature and humidity. It's only the first year, so it's not really relevant to look at the results right now. But this study will go for 4 years. So we're already seeing, that it's better in the Miyawaki forest, but I expect the results to be much more obvious in a few years. We also look at CO2 level and volatile organic compounds. That's the client is interested because he wants to see, do Miyawaki forests really improve pollution level and air quality, and also fine particle levels. That fine particle levels impact health quite dramatically. So it's quite interesting to look at that also, and you can already see that the blue line, it's outside the forest and it's way higher. So it's quite obvious that the forest is improving air quality. Thank you very much.
So I'm Prassede Calabi. I have a PhD in behavioral ecology and have been working on this fast forest for a couple of years in Winchester, Massachusetts. Some of you visited a week ago. We are a project under a 501c3 in our town. So not government, not personal, but a 501c3. And our forest is about 6,000 square feet. We have about 40 native species, about 1,100 individuals. 17 of them are trees and the rest are shrub species. And we've locally sourced everything in order to match as best possible local genetic conditions. We had intended to plant in March, but it was the third wettest March on record in Massachusetts, and it was mud pies at best. So we ended up planting in September, and it was raining in September. The community, we had over 200 people sign up for planting. They included grandmothers and grandchildren, as you see here. Some of the children we had a lot of children under 8, I would say, who were very motivated and helped a lot. This is what it is now. Most of these plants are as tall as I am, or nearly as tall as I am. This is from your site visit last week. And this is what it looks like. You can see the mulch. This was about 6 weeks ago, where you can see all the individual plants much better. That's the organic farm in the background, which has not to do with us. That's a separate plot of land. So remember that basically a forest is a food pyramid. What we're trying to do is to restore or recreate these food pyramids. You remember that from 4th grade, right? A food pyramid, and the base level is the plants, and we are focusing on planting plants. In fact, we now know that the base level is the microbiome, what we don't see, but what in fact is incredibly important to the success of the whole, because each of these layers is dependent on lateral, on above and on below. And if we don't have the integrated whole, we're not going to have an effective forest. One of the nice things about thinking about it this way is that it also helps you understand that there are tools you can use to understand each one of the different levels, some of which we've just heard about. And I'm going to tell you the ones that we're using in the fast forest. So research that is either ongoing or planned is a habitat value of simply by comparing keystone insect species diversity in this forest versus in other forests. And I have a list of tools afterwards I'll talk about, but so that's one. And we'll also assess, we have frozen soil samples to assess the soil microbiome. We're hoping to work with the professor at Boston University. Microbiome, as you probably know, is a very hot field. We have not understood much about it. We now know that ecosystems generally have a signature microbiome, and that's likely to have the same set of species, whether it's in ants or in butterflies or in birds or in mammals, as well as what's in the soil. The better we can understand this, the better we can create the kinds of networks that we need to have for successful habitat. If we have a lot of little forests spread out, how important is it that the microbiomes be consistent? Does it matter at all? These are questions that would be very helpful to know, and we just don't have the answers yet. We'll be comparing 3 different forests, our own, which is a red maple wetland forest, and a naturally occurring red maple wetland forest, which grew up by itself, no human activity, and Danahy, because that was the first one in this area, and it's a completely different provenance. And how different are these microbiomes? Are they different? What can that tell us? And how can we use this information moving forward? I certainly don't want to say, someone asked this at the site visit, I'm not saying that you need this information in order to plant a Miyawaki forest, but certainly the more information we have, the better we can characterize the environment, the more successful we can be by what we plant, because we'll know more what we have to target at a different level than the levels for which we already have information. So we are also looking at, I'm going to skip over the third one for the fourth for the moment. We have, among the many participants in this forest, there's the high school environmental science classes have built this forest and project into their curriculum, and they are doing an annual visit every year. They've already been once, twice, and they measure the diameter, the height, take a lot of other parameters on all 1,100 individual plants. They've developed a really terrific spreadsheet / map in which they know where each plant is located individually. It's an active cell. It has in it all the data, Latin name, common name, height, diameter, and condition. And so far, it looks like we have had under 5% mortality in the 10 months since we have planted this. They're going to be coming back in mid-September to do an official count, but based on our count, it seems like it's under 5%, which would be pretty great. We're also doing something which is not directly ecological. We're assessing attitudes of participants in the fast forest. It's kind of a great controlled experiment in so many ways. And John Coley at Northeastern University has done a lot of work on eco-anxiety. I'm sure you're all familiar with that. And we know that in general, action is the best antidote to anxiety. And in particular, can you see any difference in people's anxiety or emotional health in general if they have been involved in planting or working in the fast forest? The data that we collected was before planting day, and this literally as people were coming onto the site and as they left the site, and follow on a month later. And those results I look forward to. I don't have those yet either, but it's a nice add-on, especially if you're working with kids. You can see that they're happy, and that's really a good indicator right there, but it's also nice to have some formal assessment of anxiety. I should say that we had 200 people signed up to plant, and we were so efficient because we laid the plants out where they were going to be planted on a day prior to planting. We were so efficient, we finished 3 hours earlier, which gypped a lot of people from planting, and they were pretty annoyed that they hadn't been able to plant and disappointed that they hadn't been able to plant. So the kinds of tools that we've been using and that we mention, in fact, I should say, I developed a 4-page schematic how to do a Miyawaki forest, which I'm happy to send to anyone who's interested in it. Shows you what we did and the tools that we used along the way. Obviously, we did a floristic quality index. This is an algorithm that's been developed by botanists and ecologists to talk about the habitat value of any given plot of land and the plants on it. We compared ours to adjacent land. I'll get to that. I'm going to give you the overview of the tools and then some of the results. Insect biodiversity comparisons between our site and other sites. The EWA Buggy app, for those who don't know it, is really terrific because it allows people to collect data who haven't necessarily taken courses in entomology and insect ID. It has an iNaturalist type approach, so it's a very effective and very accurate way of collecting data. iNaturalist also has a geolocate function where you can identify by using GPS a plot of land, and any photographs that are taken are automatically located to that land so that you can easily collect data without even knowing what things are, just taking pictures of them. Drones, we've just seen a couple of really great examples of how you can use drones. We're very lucky that a participant in the project has one for her own work, and I'll show you that as well. I mentioned the spreadsheet that the students developed, which is a literal map where every plant is in real space where it is in the forest. We used a trail camera because we were worrying about herbivory by particularly vertebrates, but also we just wanted to know what was going on in the night. It turned out to be a really powerful tool. They're quite inexpensive. And of course, Homegrown National Park, map your forest. So this is ours about 6 weeks ago. It's filled in substantially since then. This is a drone picture. We're finding she has a close-up camera and that we can actually identify a lot of the plants off the aerial photographs, which is a very nice tool, particularly for the students who don't have unlimited time in the field. They come out once a year for a 4-hour session, but there's only so much you can do in 4 hours. So this is our workaround for that. They can use these back in the classroom. Our floristic quality index, Walter Kittredge worked with us on this forest, and this was part of his contribution. We assessed the habitat value of what was on the site when we started. It was sort of agricultural, fallow agricultural land, had a lot of goldenrod, which we know is an excellent plant, but its overall habitat value was very low because lots of things don't eat goldenrod. It's only good for some species. There are also forests. If you were there, you'll see that there's some kind of scruffy forest around the site that has a habitat value of about 13. And the fast forest site, this algorithm includes invasives, aggressive species, woody plants, their longevity, and a few other parameters. What we planted on the fast forest has a floristic quality index of about 30, which was a very nice thing to be able to say to people, yeah, we know what we're doing, and this is in fact going to give us a lot of habitat. Here are some of the things we discovered with the night camera. These are fisher footprints, coyote, fox, tiny little mouse here. We also have a couple of deer looking right at the camera, which is outside the exclosure, but I think the noise must have attracted them. And when we started, we didn't have enough rabbit fencing, exclosure fencing, and we found rabbits in the exclosure. I'm happy to say that we haven't had any since we have 2 layers of electric fencing, 5 wires on one, and we have 1/4-inch mesh and quite a bit. But this gives you excellent data, and it runs for weeks and weeks on 8 batteries, 8 AA batteries. So you can really tell what's going on. And again, you can do a comparison between this and other areas. So that's a quick trip through our fast forest. Please get in touch if you're interested in the how-to, our 4-page what we did and all the resources that we used, or have any other questions, please feel free to get in touch.
Hello, everyone. So my name is Melory Brandao. I'm in Dr. Talamy's lab at the University of Delaware. I'm a master's student. And my project is about assessing insect diversity and herbivory in Miyawaki forests and other urban tree contexts. This is my first season of fieldwork, so I don't have any cool data like my colleagues who presented before me, but I'm just going to tell you about what my study is and how it's going so far. So next, please. So for background, I know Doug definitely talked about this. Insects drive pollination, decomposition, food web. They are essential to our ecology. And it is very difficult to be an insect in an urban environment. They're impervious surface, non-native plants, what have you, but there's also an opportunity because of projects like this, breeding projects can be an opportunity to increase biodiversity for insects. It's proposed that Miyawaki forests are helpful for biodiversity, but there's very little studies if they're ecologically beneficial to insects. So that is where my study comes in. So my objectives are 3 of them. I'm going to compare insect herbivory and insect abundance, as well as diversity across 3 tree planting contexts. So our Miyawaki forests, park trees, planted managed trees around parks or around cities, not necessarily street trees, and remnant forest patches, so interstitial forests and volunteer trees. My second objective is to track seasonal changes in herbivory by monitoring leaf damage on the same branches over time, and then to evaluate how planting context influences tree-insect interactions among these different planting contexts and among different species. So I'm going to look at, or I am looking at 6 different Miyawaki forests across the Boston Metro West. They range from 1 to 4 years old, Dennehy Park being the oldest one at 4 years old. And for each site, there are comparison park trees and remnant forest sites. So for each Miyawaki forest, we had to go find some remnant forests or some planted park trees to match the species in our Miyawaki forest. So we're doing 3 rounds of sampling this summer. We finished our first sampling round last week. We're starting a new sampling round this week, and then hopefully mid-August or end of August, that'll be our 3rd round. So our focal species are red maple, red oak, and black cherry, just because those support a lot of insects or most insects. Please. So this is the Metro West. Boston is there on the right. So our sites are Dennehy Park, Somerville High School, Green Rose Heritage Park, Brookline Playground, which was not planted by Bio4Climate, but the site it's a really full forest already, and I thought the planting was like similar enough that would be good to study. And then we have Natick High School, which is in the suburbs, and then all the way on the left is the McGrath lot in Worcester. Next, please. So I had a plan for my study, when I was back at school, and me and my advisors came up with this plan. And once I actually got here and took a look at these Miyawaki forests, I realized we had to throw that completely out of the window because none of us had ever really seen. I'd seen one once before, but it was like the Somerville High School one, which was very thin at the time. So I didn't realize that our original study plan would not work. We were originally going to use beat sheets, which is like a square meter white sheet in which you like whack a little tree and like the bugs fall onto the sheet and you count them that way. And then once I got here, I realized that there's no way that's possible. All these trees are just like so close to each other and it's just a mess of trees. So I got the species, the diameter at breast height, and crown light exposure, which is a scale of 0 to 5 on how much light the crown of a tree gets, and their health. This made it easier to identify our focal trees for repeated sampling and mark them. Then we would search surrounding neighborhoods to locate comparable park trees and remnant forest patches. Sometimes we would use the city's tree map. Sometimes the city's tree map is non-existent or not helpful really at all. So really the most effective way to find these other trees was just to walk around a whole bunch. So doing the site inventory provided a baseline for these ecological comparisons across all these sites. So far we've inventoried almost 900 trees and shrubs of like 103 species across all sites, which is incredible to me. Slide, please. So this is Green Rose Heritage Park. This is our perimeter tree inventory. Excuse me. So me and my one technician, we just did this to all the sites where we just looked at a meter into the forest and inventoried everything that we saw, literally everything. So here's Green Rose Heritage. It's about 96 trees around the perimeter consisting of 26 different species. I thought about, listing the species on the legend, but it was so crazy overwhelming how many species. So I was just like, okay, focal trees, non-focal trees. Next, please. So the first part of our surveying is the insect sampling. So we're doing what we call total search, which is a 3-minute timed search per tree, where we do a full inspection of the foliage, the stems, the trunk, and we identify the insects that we find to their lowest taxonomic unit that we can identify, or we take a picture and put it or send it to an entomologist or put it on INAT for later ID. And this is much easier than a beat sheet, and it's repeatable at all sites and, across different planting contexts as well. Next, please. And then the next part is herbivory sampling. So we look at 30 leaves per tree that we inspect, and we inspect it for percent herbivory damage. So it's by the 5% increments. We try to note the damage type if we can. So mandibulate, haustellate, leaf mining, galls, etc. I'll show you in a second what those look like. And we're tracking damage over time and across tree species. So we did one herbivory sampling at the beginning of the field season, and we'll do another one at the end of the field season. So to reduce bias in herbivory sampling, we actually use this website called the Zacks Herbivory Trainer. I highly recommend it. It's also just very cool. It's basically gamifies trying to guess how much herbivory damage there is. It's to the 1%, which is like, you know, we don't do to the 1% because that would be a little too accurate. But yeah, highly recommend if anyone ever has to do an herbivory damage assessment. Go ahead. So some of the types of herbivory, we have mandibulate, which is, you know, biting, things like caterpillars, beetles usually have mandibles that bite. We have leaf mining, which I'm trying to, I can't think off the top of my head what insect leaf mines. But basically they insert themselves into the tissue of a leaf and kind of make a tunnel in through. And then we have haustellate, which is piercing and sucking. A lot of aphids do this. Next, please. And we're also taking note of leaf roll, which you know, a lot of caterpillars do, and galls, which are formed, which is a plant reaction to an insect either laying an egg or doing herbivory on a plant. So these are, I think, cherry gall wasps, or cherry gall wasps or cherry gall aphids. I'm not quite sure, but those are the types of herbivory that we are looking for. Next, please. And yeah, so this is my first field season. I will also repeat this again next year so that we have just a better collection of data. And then after that, I'll be able to analyze insect richness and herbivory trends and the like top-down effects of plant-insect interaction. So if you have any questions or want to collaborate with me, like please feel free to email me. And I want to say thank you to Bio4Climate for hosting this, and thank you to Dr. Talmy as well.
So yeah, I guess my first question, you know, is around data collection methods and tools, but also about embodied knowledge and how you as scientists, when you begin to interact with the site, make decisions about your data, what data you're going to collect, but also like the systems you're creating around data collection. And I wonder if you can speak to, you know, the Miniforests community is, there's also a lot of citizen scientists here that want to begin their own data collection methods. So I wonder if you can speak to that, like how you take these decisions, how you keep track of your data, how do you prioritize data, like the subjectivity versus objectivity component, and that is in contrast to what Kawaki was talking in his presentation yesterday about how you actually connect with the forest and form this relationship. So I'll just start with that. You know, you can improvise any answer, and then I'll go into the nitty-gritty questions that are in the chat.
I think Kawaki, what he said yesterday was really on point. Like a lot of the places that and the green spaces, I think, and for me, like when I measured trees, I've spent a lot of time out in the field, in person, the same places, trying to really like I think there's a lot. I come from geography, and so we have human geography and physical geography. And so on the physical side, you know, we measure things, we take the temperature, we're using instruments, we're trying to build up repeatable field studies that, you know, can make more generalizable types of knowledge that other people could use. But I think on the human geography side, and when we've done interviews and when we've had field journals and kind of like taken more account of like our own lived experiences in these spaces, like a lot of the reasons why I was drawn to the Miyawaki Forest method were from time spent in people's backyards and with thousands of trees over time. And those kind of like intuitive lessons, I think, are also valuable and shouldn't be just thrown out. So I'm very like, there's no bad data. All data is good data. And there's many different types of data that we should kind of like be open to. So I would encourage, especially with citizen science, like be consistent, try to create protocols, but also just go back. I liked what Kwaki said, like get to really know a place, keep going back to the same place, and spend a lot of time in those spaces.
Thank you so much, Nick. Does anyone have anything to add to that?
I just
Uh,
want to, oh, sorry, you go first.
I go. You have to be. I mean, if it depends, how much time and material you have. But in my case, I had to be very simple. It had to be effective. So, for example, water infiltration rate to me is a very easy indicator to take. Anyone can do it. And to me, it gives a very good idea of soil quality. So, uh, yeah. So don't be impressed, and also just spending time, around the forest, looking at it. Um, you learn so much, and the good thing also is you talk about it much better, because somehow people feel that you have the experience, and the way you describe things is more convincing.
Yeah,
I
that is.
was going to say that, yeah, I totally agree with just spending as much time at your site as possible. As somebody who had to look for a lot of, reference sites, park trees, and interstitial trees, a lot of cities do not have up-to-date tree maps or tree inventories, which made it really frustrating for me. So literally just walking in every direction all around was just the most helpful thing to do, which anyone can do. And, we didn't use any kind of fancy like data tracking technology. We literally just used the Trees Count app, which is by the Texas A&M. Anyone can download it. It's very easy to keep track of trees that way. So yeah.
It also I found that especially by having so many people participate, we had the list of organizations includes churches, schools of all sizes, kindergarten groups, Girl Scouts, Boy Scouts, different, faith ministries. There was the experience of being in the forest and people saying, even planting in the rain, oh, I haven't played in the mud since I was 6 years old. I can't believe I get to play in the mud. And it has some carryover to whatever else they're doing, and it kind of changes their experience of the world. Now, somebody told me that when she looks out at her yard and she sees what I call dead green, it's stuff that has no ecological value. And she was going to go buy an ornamental plant. And then she said, well, I heard your voice and I asked what would be a native. And there's a carryover. I like to say it's 51% ecology and 49% community, because without the community part, you're never going to get the coverage that we want and get the private land turned over. So the experience, the sharing it, the teaching, I think is a really, really big component of it all.
Thank you so much. Yeah, that's incredibly insightful. And I guess like a quick follow-up question would be like, for those who are interested in designing their own protocols, if you haven't mentioned it already, what would be like an advice that it's still, rooted in science, but doesn't need all the tools that a scientist has? Like what would be a first step? Or yeah, I know for me, and for my other scientist friends, we talk a lot about like keeping track of data and always interacting with that, but like that is the hardest part. So yeah, I wonder if you have any advice of designing these protocols for, and also I think that's connected to a question I had around critical research gaps and like, I don't know, like if you can speak a lot about like in your own field, what are some research gaps you're encountering, especially when it comes to mini forests?
I think there's a lot of tools out there. It used to be a huge impediment to doing citizen science was that people needed to have a lot of expertise. You couldn't just take a class outside and expect them to collect meaningful data. But now between iNaturalist, which some people worry about students using because they worry that student location information will be transmitted and that that's not legal in some states for student protection. But there are things like the buggy, the EWA buggy, which doesn't do it geolocates to the study site, not to the student. So I'm having that argument right now with the high school. So there's a lot of things that are available. Anybody can use Excel, anybody can use mapping programs. There's of course GIS, which is more complicated, but the availability of tools is just exceptional. And even being able to take photographs. When I started teaching, I had to get slide rules to get people to do computation. Everybody has this high-powered iPhone that can do all kinds of calculations and even some statistics and so on. I feel that the important thing is to be systematic about it. And however, if there's somebody who's really good at it or who has even good handwriting to take notes or something, just be systematic and maybe don't get too big too fast. Keep it something you can do so that you don't get depressed about it. Don't collect data every single day if that's not reasonable. Collect it once a week or once a month, but then really do it. And even we have an elementary school group that's doing photo data collection on how quickly the forest grows with how quickly they grow. So they're taking pictures of themselves in front of the forest, and they want to do this every year through high school. And I keep telling them it's going to be here for a couple of 100 years. You come back from college, you come back to get married, whatever, you can go and take a picture there again, and you can have this personal series, which also gives you some data. So make it fun, make it not just nitpicky. And I think that part was, I did not expect, although I teach and I know lots of students get excited in this, I didn't realize the amount of enthusiasm there would be by the populace at large. And that's been a real treat. And so trying to turn that into keeping people engaged and doing that kind of even low level data collection, it's not only meaningful for them, but it's real data.
Yeah, I'll say that I've had this experience over and over again. I also have a floating wetland project here in Providence, and just visiting it regularly and observing the plant growth and asking all these questions, and but also like finding one tree that I always visit and see what changed, like continuously finding or like establishing a relationship with something in your environment that you can visit over and over again. It causes this beautiful inner transformation and cultivates this sense of belonging that I really, really, really love. And that's one aspect of mini forest I appreciate. Anyone else wants to add to what my question and what Prassede said?
I think on the temperature side, there's, in terms of the, I'm very interested in both different times of the day. So you saw like, we know that the forest is colder than like a cement parking lot, but like how much colder, how will that change over time, both in terms of years, but also how does that change at different times of the day? I'm very interested in how land surface temperature interacts with air temperature, with humidity, with feels like temperature. And then how far a Miyawaki forest effects might be felt. So if you're in the middle of the parking lot, does the Miyawaki forest make a difference for you? And how close do you need to be? I love the Doug's kind of like classifications of plants that contribute or don't contribute or are disruptive. So like, do those contributors also, are they cooler? Are they providing more cooling if they're in a native location or if they're in a community versus alone? So those are all questions that keep me up at night, and looking forward to continuing working on them.
I feel like you guys answered those questions really well. I don't know if I can add.
One question is actually from Nicolas, Brabandère. Why is the maintenance not always needed in the third year? What were the results that led you to that conclusion? And sorry to
It's really Professor Miyawaki, that tell us that a Miyawaki forest should be self-sustaining after 3 years. If not, there's probably a problem somewhere. That's also very interesting, maintenance is so costly in cities. And so if we can bring a solution that doesn't require maintenance over the long term, it's very strong arguments for many of my clients. So my experience is I can see that I have to do maintenance the first 2 years, and usually later on, the trees are big enough, they grow over the weeds. So obviously there's no need to deal with the weeds. There's no, it's just, you can see it, yeah, and also what I learned in Japan is if you overdo the maintenance, it can have a very negative impact on your forest. It might not look like this for most people. They might be happy that we've done some maintenance, but sometimes it can really, when you do a Miyawaki forest, you're looking for the wide, so the wide aspect of it. So if you do too much maintenance, you lose that. And sometimes the natural growth of a tree is troubled by too much maintenance. If you trim branches and stuff, they don't grow the way they should. And sometimes instead of growing tall, they grow wide, and then they take the light of the neighbors. So, Miyawaki is very clear. No management is best management in Miyawaki forests.
Yeah.
I do
Yeah.
want to add one thing to that.
Absolutely.
So we are having discussions about whether to replace things that have died. There's one species that's clearly too wet, they all died. That's one piece of it, but the piece we watered when we planted because we wanted to be sure they would establish. We haven't watered since because that's part of the match between your list and your environment. And we have been doing some weeding, but the thing that we really cannot do without is the exclosure, because the rabbits at this point would have destroyed everything. And I don't think that is counter to the Miyawaki philosophy. I think that's just being practical. And I think the fence will be there for years because it's also next to an organic farm, which draws rabbits probably. So, it's kind of it's a two-edged sword. And we are under so much pressure, ecological pressure due to absence of habitat, that within the paradigm, if we can ensure that these trees survive, it's a huge amount of effort. It's a lot of work and money to plant and to, why would you take away the fence in year 1 or 2 or 3 if you know that you're going to lose most of your trees? That just doesn't make sense. And I'm sure that that is not counter to the Miyawaki philosophy. We're not doing anything. It wouldn't be unnatural if this was where there were enough predators to eat the rabbits, right? Then it would be a balance, but there's not a balance. We've so unbalanced things that we need to work with the unbalance up to some degree so that we can get things back into balance. And I feel like the rabbits is a good example. They're just completely out of control. There's not enough predators.
Yeah, thank you for offering that perspective. And also, like going back to water usage and watering the mini forest, I feel like also what Dr. Akira Miyawaki is suggesting with no management is best management is like emulating nature and how nature, like the rainfall, the irregularity or rainfall, or like completely relying on it. And I think in our human systems, something I think a lot about is, yeah, this need of control and putting on a timer and watering regularly. And that's just not how nature would provide water to the plants. So that's very interesting to me. I don't know where I sit in that yet, but I've been taking this water cycle restoration class with water stories, and I feel like my entire perception of the water cycle is shifting right now. So, yeah.
But to be clear, we only watered when we planted them because it was late in summer, and once they're established, we haven't watered at all. And in fact, I can say this, we have 600 feet of hose in 100-foot sections, which we are happy to loan anyone in the area if anyone needs this when they're establishing whatever they're planting. Please feel free to get in touch because we're happy to loan those.
Yeah, thank you. Yeah, and I guess we have time for just one more question because we went 10 minutes over time. We're just gonna take 10 minutes from the break. That's okay, I hope. I think actually this question is for you, Prassede. It's around, it comes from Jared, and Jared asked that you mentioned that the trees in the Winchester mini forest were sourced locally for genetics. What does that mean, and why is that important? Yeah.
So one of the things that's hardest, I think, for people to wrap their heads around is that you not only need native species, you have to have a really good match between the environment in which your forest is and the species you put there, because you don't want to do maintenance. And if you don't have a good match, you're going to have to coddle them in various ways, and it's going to be pretty frustrating. To take that to its maximum, you want genotypes that are local. And if you look at, for example, a forest, they grow their own seeds from they grow seedlings from local seeds to be sure that these are local genotypes, because you know that those are the ones that have been selected over time and are going to be the best match to the area where you're actually planting. We certainly don't want to spend the time, and the labor is a big issue to grow your own seedlings. So we sourced from a nursery that grows their own stuff, and that is an hour's drive away. So we were pretty confident that the genetics were pretty similar to what is around here, which increases their survivability and also probably makes them a better match for the local insects. Sort of the more co-evolved potentially these things are, the better they are, the more likely they are to succeed. So we wanted to do everything we could to help this be as natural as possible. You know, who was it who said, what would nature do? That's always my question when people say, oh, I don't know if I should do this or that in my garden. And I say, well, what would happen if you let it go? What would happen if, and so naturally what would be growing here is all the stuff that's blowing in and brought in by birds and so on. So we're trying to mimic that as nearly as possible by using things that are from the area.
Thank you so much. I guess like as we're wrapping this, I would like to go back to my like initial question of, but maybe like not asking questions, but more, I wonder if all the presenters today could offer, as we're wrapping up, like a 1-minute reflection of what you've learned from each other today, or what knowledge gaps research gaps you would like to fill in, or anything you want to share as a closing thought. And I think, yeah, one minute each because we are over time. I'm sorry about that. It's, we could definitely go on and on and on. And I hope we will in the near future.
I can start. I'm really excited to go visit the Winchester farm with my drone co-pilots next week, and hopefully, I think like it's amazing to see how many Miyawaki forests Nicolas has looked at in in Europe and Belgium and France, and building out that network here. This makes this conference so exciting. Melory, the findings that you have around herbaceous. What's going on with the leaves? I'm like, is there any connection with temperature? I would love to like collaborate on that. And especially all the tree monitoring you're doing, because I'm just like, it's a forest, but you're actually seeing the species. So yeah, just really excited about both the representative, like the future. Hopefully we can get to what Nicolas has done in Europe here. And this network is a great start.
Anyone else? Closing reflection? You don't have to. That's totally okay. And yeah, Nicolas, I'm so grateful you were able to join us from Belgium, all the way from Belgium.
Thank
Yeah.
you.
Yeah.
I just want to say to Nicolas from Belgium, I saw your report when I was like writing up my proposal, and it was the most in-depth, report about biodiversity in Miyawaki forests. It was very helpful while I was doing my research, and it's very cool, and your data is so interesting, and I'm really excited to work with you guys. I would really love to see the Winchester Forest. If you're doing drone searching next weekend, I'm also free next weekend. if you
On
wanna
your
do-
own. That's true for anybody. Please feel free to get in touch. Love to have you come visit, pull up a few weeds, hang out, whatever.